Okay. So, heredity and evolution. So, heredity and evolution means what? First of all, heredity means something which is passed on from one generation to the another generation. That is, you can say from the parents to offsprings, whatever characters are being passed. This is termed as what? Heredity. Whereas evolution means what? Evolution means a particular organism. For example, if we take the ancient organisms which are there, they have been evolved. Evolved means some of their parts which are there. For example, it may be the forelimbs, hind limbs, tail. Okay. So all those stuff has been what? Modified, and they have been evoluted into a entirely different organism. So this is called as what? Evolution. So heredity means what? Whatever traits. Traits are only called as what? Characters. They have been passed from parents to offsprings. Whereas evolution means what? It's a process in which certain parts or organs are either, you can say either diminished, okay, or suppressed for producing different organism to survive. Okay, so this is what heredity and evolution. So first of all, let's go to the heredity. Okay, so now as I told you, it is nothing but the characters. It deals with what? The characters or the traits. So if you consider a trait, basically they are of two types. Okay, one which is called as acquired, and another one which is called as inherited. Okay, so characters or traits are basically of two types, that is acquired and inherited. So now what is this acquired and inherited? Acquired means when a person is born or when an organism is born, during his lifetime, he is going to get that particular character. So that is called as what? Acquired trait. Means which an individual can experience during his lifetime. Okay, so from the birth, it is not present. So during the lifetime only, he or she will achieve it. Whereas inherited means what? It means already from the ancestor, that person or that her or him will be having that particular trait or characteristic. So here, that one will acquire from his or her ancestors. Okay, so because of this, whatever the acquired traits are there, here, what do you say, non-reproductive tissues are being involved. Okay, in the acquired trait, non-reproductive, because they are not genetically passed, so non-reproductive tissues involved. And because of that only, they cannot be passed onto the offsprings or progeny. Okay, whereas inherited will be opposite. That is, over here, it will involve what? Reproductive tissues. Whereas it's also transmitted to progeny. Okay, so even though there are certain characteristics which are being passed, okay, but there are also certain characteristics which are achieved during the lifetime. So due to this, one more term comes over here, which is called as what? Variation. In simple terms, variation means nothing but differences. Okay, differences among whom? Differences among the individuals of a species, and also you can say among offsprings of same parents. Okay, like for example, if you take flowers, okay, so in the flowers you cannot differentiate that this flower is different, or like for example, if you take a rose, okay, all the roses look similar. But in the same way, if you take the examples of say human beings, okay, whatever parents are there, alike the parents, the offsprings will not look the same. Correct? So that is only called as what? Variation. They are not cloned. So there will be certain variations of that particular offspring or the progeny which is produced from the individuals. Okay, so that particular thing is only called as what? Variation. So from the same parents only that offspring will come, but they will be having a variation. So you can identify them easily. Okay, so this particular heredity or the variations which are there, either the variations may be like what do you say, heritable or non-heritable. Okay, means either they have been passed from the ancestors or they may not. Okay, so according to that only we got this variations in individuals. So on the basis of the heredity only, there is a law given by a person, that is Gregor Mendel, and that law is called as Mendel's law. So he had generally given three laws. Okay, so now we are going to see that some of the terms which are related to the Mendel's law or the Mendel's work, you can say. So as I already told you, depending upon the heredity as well as non-heredity characters, variations occur, right? So whatever variations are there, either you take it maternally or paternal, means father or mother. That all will be influenced by whom? The paternal and maternal DNA. And DNA is only the hereditary material which is present, okay, which makes up this variations. Fine. So now this particular person, that is Gregor Mendel, he had done some of the investigations or the work during his era. So basically, he proposed a theory which is called as Mendel's law, which states what that whatever this heredity is there, this heredity is being controlled by who? Something which is called as factors. Okay, so according to his theory, he says that whatever heredity characteristics are there, that has been controlled by whom? Factors. Now, what is this factors? Factors are nothing but genes. Okay, and what is genes? Genes are nothing but those things which takes or carries what? DNA or RNA, the particular genetic material which is important for the survival or variations or heredity, we can say. Okay, so to prove this thing only, he did an experiment. Okay, so for the sake of the experiment, he took a plant which is a simple pea plant, which is also called as Pisum sativum. Okay, so he took a simple pea plant because of its multiple benefits or characteristics, which could make him a little more easier to do his experiment. Because pea plant, if you see, okay, whatever the reproduction rate is there, that is also greater. Okay, variations also you're going to get more, as well as it is available all the time. Okay, so many characteristics are there of the pea plant. So it was an ideal plant for his experiment. So he used this pea plant for his theory proposal. So here, after using this pea plant, he used what? 7 contrasting characters. 7 contrasting characters. Okay. so that he could study this particular thing called as heredity. Now, in this seven pairs of contrasting characters, you have different terms. Okay, so first let's see some of the terms. So the first one is the dominant trait. Trait means nothing but characters. Okay, so basically we have two types over here. One is the dominant and second one is the recessive. So the first one is dominant trait. So dominant trait means what? Those characters, okay, which will be able to express itself over another contrasting traits. Okay, so dominant trait means what? It will be dominant, means even though if many other traits are present, that particular dominant trait only will be expressed, suppressing the other characters. This is called as what? Dominant trait. Okay, means it is going to rule over the other characters in short. Whereas second one, which is the recessive trait. Recessive trait means what? The opposite of dominant. That is, it is unable to express its effect in the presence of dominant trait. Okay, for example, dominant trait, you can say it's a capital TT. T stands for tall. Okay, and capital TT means tall. Whereas recessive means what? Small tt. Okay, which means short. So this is for the tall and this is for the short. So in this, like this only, many characteristics you can take. It may be the tallness or the, what do you say, the breadth, the color, okay, whether it's wrinkled or round. So many characteristics are there. That's why he gave this seven contrasting characteristics, out of which one such characteristic is what? Tall and short. So tall, dominant means what? It will be capital. Whatever trait you're going to take. For example, if you take yellow and green, okay, maybe yellow you can write as capital YY. Green you can write it as what? Small YY. Correct? So here, if this capital Y is present along with the small YY, the thing which is going to get expressed is what? This capital YY. Means even though if you are having a green and a yellow, who is going to get expressed according to the law of dominance or dominant trait? The green is going to get expressed. Okay, so this is only the difference between what? Dominant trait and recessive trait. So next term is the homozygous and heterozygous. So homozygous and hetero. Homo means nothing but similar. Okay, so whatever characters, factors, genes, whatever is there, they will be similar. For example, you can take, say, capital TT or small tt. So if you see, both are similar. Okay, but if you take the heterozygous, means what? It will be different. Means whatever the traits are there, it will be different. So instead of capital TT or small tt, what you can write? One capital T, one small t. Okay, so this is nothing but tall. This is short. Whereas here one capital T is there, so dominant. So this is also tall. But homozygous and heterozygous is according to how you're writing this particular thing. So if both are capital, both are small, it's homo. If one is capital, one is small, it's hetero. Okay, next term is what? Phenotype and genotype. Okay, so phenotype means the external characteristics. You can say it as morphological characteristics. Okay, so easily, like it can be identified. For example, tall, short, okay, or yellow, green. So easily by looking only, you can see the differences. So that's called as what? The phenotypic things. Okay, so it is nothing but observable. Observable. For example, I told you tall, short, or color, whatever it is. Whereas genotype means what? Genetically means you cannot see it through your eyes. Okay, only when a particular investigation or experiment is done, then only you can identify the thing. So hence, it is nothing but the genetic constitution of a organism. Okay, for example, if you're going to cross a tall and a short, after crossing only you will know whether you're going to get a hybrid ones or whether you're going to get either tall or either short. Correct? So only after doing that experiment or investigation only you're going to get that particular trait or character. So hence that is called as what? A genotype. Okay, for example, when you cross a tall and a short, that is capital TT and small t, maybe you're going to get what? All tall. And that tall will be maybe pure tall. Okay, so all will be TT maybe. Okay, so this is only called as what? Genetic constitution or the genotype. So according to whatever crossing you're going to do between the two contrasting pairs, you will get a particular ratio, okay, which is called as what? Genotypic or phenotypic ratio. Phenotypic ratio. So genotypic or phenotypic ratio means nothing but whenever you are going to cross two particular contrasting characters, you're going to get an expected ratio of that particular thing. Okay, so that is only called as what? Phenotypic or genotypic. Means in short, you can write it as expected ratio of genotypes produced by particular cross is called as what? Genotypic ratio. Whereas instead of the genotype, if you write the phenotype, it is called as what? Phenotypic ratio. Okay, so most probably this phenotypic and genotypic ratio depends upon two more things. Okay, that is nothing but the cross how you're going to do. Okay, so basically two types of cross are being there. That is one is the sorry. Yeah, one is what? Monohybrid and the second one is dihybrid. Okay, so mono means single, di means two, and hybrid means whatever offsprings or progeny at the end you're going to get after crossing is what? Hybrid. Okay, so depending upon whether the cross is monohybrid or dihybrid cross, you're going to get this, what do you say, expected ratio of the phenotypic or genotypic ratio. For example, monohybrid cross means what? Because it's single, so only over here, one pair of contrasting characters are being involved. Whereas in dihybrid, what will happen? Two pairs will be involved in crossing. Okay, so if you consider a phenotypic ratio for the both, for example, monohybrid and dihybrid, then you're going to get over here for the monohybrid. Okay, this will be same for every crossing. So if you're doing a monohybrid cross, then the phenotypic ratio will be fixed, which is 3:1. This is fixed. Okay, whatever, irrespective of whatever characteristics you take, 3:1 you're going to get. Whereas if you're taking a dihybrid, then whatever the phenotypic ratio is there, that will be 9:3:1. Okay, so this will be fixed. So in the exam, if they are asking what is the phenotypic ratio of a monohybrid cross, irrespective of whether you're taking the tall, short, colored, or whatever thing, it will be 3:1 only. Okay. So after crossing only, what you're going to get, the first thing is the phenotypic ratio. Genotypic ratio is difficult, mostly in case of the dihybrid cross, because you're going to get a very big, what do you say, hybrid offsprings. So it is not possible to count them all. Okay, so hence every time phenotypic ratio is being considered. Okay, so as I told you, this particular Gregor Mendel, he did an experiment by taking the pea plant. Okay, so initially when he started this experiment, these are the terms which he used for the experiment that we are going to see now. So first, let's see what are the stages in his experiment. So the first thing, or the first stage of his experiment, was what? Selection of the parent plants for the crossing. So whatever parents he took, they were what? True breeding with contrasting traits. So true breeding means what? Homozygous they will be. So if it's a tall, it will be capital TT. If it's a short, it will be small tt. So he took two contrasting pairs which were pure tall and pure dwarf. So when he crossed this two, okay, first cross which he got was what? F1 generation. F1 is for filial. Okay, so filial generation. So after getting this F1 generation offspring, he crossed this F1 into F1 again to get what? F2 generation. Okay, so when he crossed this two, like if we see over here, take a T from here, a small t from here. If you cross this, what you're going to get? Capital T and small t. So according to the dominant trait, here what is expressed is tall. So this is your F1 generation. Okay, so first, whenever he did that particular cross, what he got was what? F1 generation. So after crossing a pure tall and a pure dwarf, he got a heterozygous tall plant. Okay, that is capital T and small t. So once he got this F1 generation, what he did, he self-crossed this particular thing. Means he allowed that particular plants to undergo what? Self-pollination. So that I already told you, F1 into F1. So hence it will be TT into TT. Right? So after doing this cross, what he got? This is now under the self-pollination. What he got? He got a F2 generation plant. Now once it is entered into the F2 generation, it becomes as what? Dihybrid cross. Right? So for example, you can take capital like yellow green as colors and how that particular seed or flower looks like. So you can take a round and wrinkled. Okay, so here, whatever crossing is there, I told you it's fixed. That is 3:1 phenotypic ratio. Whereas in the F2 generation, in the dihybrid cross, what you're going to get? When you cross this, you get 9:3:3:1. So this every crossing is being done by using a square. Okay, and this square is only called as what? Punnett square. Okay, so in the whenever you're doing a crossing in the Punnett square, you have to make sure whatever parents you're taking should be true breeding. Okay, means mostly they should be what? Homozygous. Then only you're going to get a perfect, what do you say, offspring for the thing. Yeah, fine. So this is all about what? Some of the terms which are used over here. So now let's go to the actual experiment by using the Punnett square, how he has conducted the thing. So now we know some of the principles of the Mendel. Okay, and what are those principles? First one is each characteristic in an organism is represented by two factors. Okay, so two factors means what? I told you, recessive and dominant. So these two factors are nothing but what? Recessive and dominant. Okay, this is the first thing. Second thing is what? Even though if you're taking two contrasting factors, they are not going to blend together. Okay, so whatever two contrasting factors are there, when they are present in the same individual, when present in an individual, they will not blend. So hence, they are not blending together. So we can take it as what? They are independently inherited. And whatever inheritance is there, that is going to occur at the cellular level. So independently inherited means what? Whatever heredity is there, that is the passing on of genes from the parents to offspring. This is going to take place at what? The cellular level. Okay, so where this in the cell, there will be nucleus. Okay, so in the nucleus only, you're having the genetic material, which is called as what? DNA. So cell, okay, cell will have a nucleus. Nucleus will have a genetic material which is called as a DNA. DNA is going to have what? Chromosome, right? And this chromosomes only express itself, okay, to give what? New characteristics. So here basically 23 chromosomes are involved in a human body, okay, out of which the one is considered as the gametic chromosome or a sex chromosome, whereas the other one which are there, it's the somatic chromosomes, means vegetative chromosomes. Okay, so these are what? Certain principles according to the Mendel's experiment. Okay, so whatever these principles are there, that is being put forward into three categories. Okay, that is, first one is the first law of dominance. Okay, second, which is the law of segregation. And third, law of independent assortment. So what is this law of dominance, segregation, and independent assortment? See, law of dominance. Law of dominance means what? Whatever that parental trait, one of the parental trait, means if you take a tall and the short, only one parental trait is going to get expressed. That is only called as what? Law of dominance. And where do you find that particular law of dominance expressed? In the F1 generation. Okay, so what is the law of dominance? Only one parental trait gets expressed in F1 generation. But when you self-cross it, then what you're going to get is both the traits get expressed. While both traits get expressed in F2 generation. So this is called as the law of dominance. Okay. Then comes the second one, which is law of segregation. So segregation means what? Separation. Okay. So it is nothing but what? When you take two alleles, then they are going to segregate from each other. When the characters are transferred from parents to offsprings. So here reproduction plays a very important role because this happens during the reproduction. So hence whatever the dihybrid cross is there, that is applicable over here, that is in the law of segregation. Because in dihybrid cross what happens is you're going to take two contrasting pairs. So when you take two contrasting pairs, like for example round yellow and wrinkled green, okay, then as I told you, you get a dihybrid cross. So whatever the phenotypic ratio is there, instead of 3:1, you're going to get 9:3:3:1. Okay. So in the same way, whatever the dihybrid cross is there, on that basis only, the third law also was proposed, that is law of independent assortment. Okay. So independent assortment means what? Like over here, whatever members will be there, which are produced, okay, they are like of different pairs of alleles assort independently into gametes. Okay, so this is independent assortment because whenever you're doing a dihybrid cross, okay, whatever, instead of a 4-block Punnett square, you're going to get a 16-block Punnett square over there. So each and every thing, like for example, if you take capital Y, small y, okay, then capital Y, small y, and then you take it like this, okay, whatever things you're going to get as a gamete will be different, means 16 gametes is going to get produced over here. So according to that, each particular character or an allele is going to express itself independently. Instead of blending together, what they will do, they will express themselves independently without getting, suppressed. Okay, so that is only called as what? Independent assortment. Okay, so in short, for example, like for example, if you go to a shop to buy sweets, okay, what you do, sometimes you choose particular sweets to put it and make it into a box. Okay, so instead of similar kind of sweets, you take different kinds of sweet and put it together. So you are going to get what? A box which is full of different sweets. The same thing only is applicable over here also. Whenever a dihybrid cross is being done, at that time, two things, that is law of segregation and independent assortment, both are being seen over here. Okay.